Some cancers fight a slow war of attrition, but the most aggressive brain cancers, they're different. They have these secret weapons. They don't just grow, they evolve, literally at super speed, coming up with endless new ways to survive whatever we throw at them. So today, we're going to pull back the curtain on two of these secret weapons, two incredibly powerful engines of mutation that make these tumors so tough to beat.
Okay, let's just start with the big question, right? What exactly makes a brain tumor, especially something like a glioma, one of the most difficult cancers to treat? The answer isn't just about its location, believe it or not. The real challenge, the real fight, lies deep inside its genetic code and its incredible ability to change and adapt on the fly.
To even stand a chance in this fight, you have to understand the enemy. And this enemy, well, it's a master of disguise. It's constantly changing its form right down at the cellular level. So we're focusing today on gliomas. These are tumors that pop up from the supportive glial cells in our brain. And the most infamous one, the one you've probably heard of, is glioblastoma. It's just notorious for being incredibly aggressive and, frankly, for its grim prognosis.
Now picture this. A glioblastoma isn't one single uniform enemy. It's more like a chaotic ecosystem, a jumble of different cells, and each one has its own genetic quirks. This diversity, scientists call it intratumoral heterogeneity, is the tumor's ultimate survival tool. It means a treatment might wipe out 99% of the cells, but it leaves behind that 1% that happens to be naturally resistant. And that's all it takes for the tumor to come roaring back.
So you have to wonder, where does all this genetic chaos even come from? For a long time, we thought it was mostly just simple errors, like the cell's DNA repair kit was just broken. But it turns out that's only part of the story. There are much more dramatic forces at play here. Imagine for a second, what if a country's own defense system, the thing designed to protect it, suddenly started working for the enemy, actively creating chaos from the inside? Well, that's pretty much what we're going to look at today. Two fascinating mechanisms where the cell's own machinery gets hijacked to drive mutations at an unbelievable rate. And this brings us to a really key concept, hypermutation. We're not talking about a slow, gradual buildup of mistakes over time. No, this is a mutational storm. It allows a tumor to evolve faster than we can possibly treat it. And we see this a lot in gliomas that come back after chemotherapy. All right, let's unmask our first culprit. It's a protein that's supposed to be one of the good guys, a guardian of our cells. But in cancer, it flips, and it becomes this incredibly dangerous villain. Meet ApoBex3B. This enzyme belongs to a family of proteins that are basically cellular security guards. Their normal job is to hunt down viral DNA, think HIV, and edit it, riddling it with mutations to shut the virus down. It's a brilliant part of our immune system, really. But in a glioma, something goes wrong. ApoBex3B gets confused. It turns that powerful editing function inward, and it starts attacking the cell's own genome. It becomes a rogue editor, just scribbling all over the DNA, creating thousands of mutations and fueling that genetic chaos we were just talking about. And look, this isn't just a theory. We can see the proof right in front of us. The red stain you see here is ApoBex3B. Just look at how much of it is lighting up the tumor tissue compared to the normal brain tissue, where you can barely see it. Scientists can catch this rogue editor red-handed right at the scene of the crime. Okay, so here's the bombshell, and you might want to sit down for this one. Research has shown that temozolomide, the standard go-to chemotherapy for glioma, can actually cause the tumor cells to make more ApoB3B. It's an absolutely vicious cycle. We treat the cancer, and the treatment itself can power up one of the very engines the cancer uses to evolve and fight back. So, if ApoBex is a relentless storm of tiny mutations, our next mechanism is a single catastrophic event. It's not a storm, it's a full-blown genomic earthquake. The term is chromothripsis, which literally means chromosome shattering. And yeah, it is exactly as violent as it sounds. In one single, awful event, an entire chromosome gets pulverized into pieces, and then the cell's repair machinery hastily glues it back together in a completely random order. So how on earth does a genomic earthquake like this even happen? Well, it often kicks off when a chromosome gets lost during cell division and ends up stranded in its own little bubble called a micronucleus. Inside this bubble, it's unstable, it's vulnerable, and it gets shredded. The cell then basically panics and tries to patch it all back together,
but it does a really sloppy job, mixing up the order and direction of all the genes. What you end up with is a Frankenstein's monster of a chromosome. I mean, in a single stroke, this one event can create brand new cancer-driving genes or completely knock out the genes that are supposed to suppress tumors. It's a way for the cancer to accomplish years of evolution in a single afternoon. So let's recap. We have two incredibly powerful forces, the constant storm of ApoBec editing and the sudden earthquake of chromothripsis. And even though they work totally differently, they achieve the exact same deadly goal. They give the tumor an amazing ability to fight back against our treatments. At the end of the day, it all comes down to creating options, whether it's through thousands of little edits from ApoBec-C or one massive rewrite from chromothripsis. The result is just immense genetic diversity. And in a tumor with billions of cells, that means it's almost a guarantee that a few of them, just by sheer random luck, will have the right set of mutations to survive whatever therapy we use. And those are the cells that survive and grow into a new tumor that's already resistant from day one. And, these mutational engines don't affect all gliomas the same way. For instance, some less aggressive tumors can actually be pushed by treatment to evolve. They can lose some of their molecular markers that were keeping them in check, and they start to act a lot more like the most aggressive forms of the disease. It just goes to show how these engines can actively steer the cancer's evolution down a much more dangerous path. Look, I know that uncovering these dark secrets about how cancer works can feel, a little daunting. But here's the thing, every single mechanism we uncover, every secret weapon we find, is also a potential weakness we can exploit. Every secret weapon has an off switch. And that is the real takeaway here. These engines of mutation aren't just problems, they are targets. If a cancer cell is relying on something like ApoB3B to evolve and survive, it becomes addicted to it. So, what if we could take that away? And this is exactly where the research is headed. Scientists now see ApoBig3B not just as a source of chaos, but as a bullseye. In their own words, from a 2017 paper, it may therefore be a potential target for future therapeutic strategies against glioma. And this all leaves us with a final, and I think, very hopeful question. Instead of just trying to kill the ever-changing cancer cells, what if we could attack their very ability to change in the first place? What if by targeting these mutational engines, we could finally disarm cancer's greatest weapon, evolution itself?